Okay, let's talk a little bit about transfer RNA and ribosomal structure and some specifics about how the two are going to interact so that we will understand how translation is going to happen. So transfer RNA or little tRNA is a single strand of nucleotides, just like all RNA is. Only there is hydrogen bonding between the nucleotides, and that's going to cause folding. And in the folding, we end up with this clover leaf structure that has like these three different loops on it. So we have this clover leaf, and in this middle loop, what happens are there three of the nucleotides are looking to the outside, and they form what is these exposed nucleotides form what is called the anticodon, not a codon, but an anticodon. So remember, the codon is on messenger RNA. It's a triplet of bases on messenger RNA. And the anticodon is this exposed triplet of bases in the middle of transfer RNA. Now, up here at the 3' end of transfer RNA is where a specific amino acid is going to attach. So each transfer RNA carries a specific amino acid, and it is coded for it. So this is how transfer RNA is going to be translating the amino acid language into the nucleic acid language. So we're going from a nucleic acid language of DNA and RNA into a protein language by bringing an amino acid. So as I said, each transfer RNA has one anticodon down at this end, and the anticodon is the matching sequence for a specific codon on the messenger RNA. Okay, and so each anticodon has the attached amino acid to the messenger RNA codon. So if we go back and look at this one, AAA, we go AAA. My glasses on to make sure because it's tiny. Yes, AAA on our codon wheel codes for lysine. And so the anticodon for AAA would be UUU. So this transfer RNA has the anticodon of UUU, but it is bringing the amino acid lysine because lysine is what was coded for by AAA. Okay. So now, what does that have to do with anything? Well, the messenger RNA is going to be attaching to the ribosome. And so the ribosome is where the codon is going to be binding to its anticodon, which is bringing the amino acid. Okay, so here we have the small subunit and the large subunit. Here's the messenger RNA attached to the small subunit. Here's the incoming transfer RNA carrying the amino acids. Okay, so as you recall, we had our start codon of AUG. So we have a transfer RNA with the anticodon of UAC, bringing the amino acid methionine. Okay, and then we have our next codon, UUC, bringing a transfer RNA with the anticodon AAG and the amino acid phenylalanine. Okay. So looking specifically at the ribosome, in addition to the two subunits that are formed in the nucleus and, as we say, exit to the cytoplasm and join together, where we're going to have the small subunit binding to the messenger RNA, the large subunit has a real purpose. As you can see, it's got three little areas marked EPA. the Environmental Protection Agency, in that order. But remember, messenger RNA is read, and all nucleic acids are read from the 5' through the 3'. So it's actually moving in this direction. So we're actually moving from the A side to the P side and then to the E side. So we're not reading this direction, we're reading that direction. Okay. So the A side binds first. A stands for the add site. And from there, the transfer RNA will be moved to the P site, the protein site, where the protein chain is getting longer and longer. And then it moves to the E site, which is where the transfer RNA exits. Okay, because the protein stays in the P site, and then the transfer RNA without any attached amino acid exits from the E site. All right. So as it's moving along the messenger RNA site, the only one that doesn't go to the A site is the very first one, AUG, with the very first amino acid methionine joins in the P site. After that, all the other adding transfer RNAs will be added from the A site. The new transfer RNA with the amino acid will add in there, and then their proteins will be taken off. The protein chains will be taken off and added to that. The transfer RNA gets shifted over to the E site, and it gets kicked out from the E site. The whole thing gets shifted down so that there is a new codon in the just below the A site, so a new transfer RNA can be added in at the A site and bringing a new amino acid with it. So take a moment, name the 3 ribosomal sites, what happens in each site, go through the structure of the transfer RNA, go through the difference between a codon and an anticodon, what does the amino acid on the transfer RNA match, and go through what the special codons are. I want to thank you for all your hard work, and then when we come back, we'll be ready to talk about translation. Finally.